Invert mix the blood tube well before use. Add 400 microliter blood into 1.5 ml sterile microcentrifuge tube. Then add 1 ml of blood lysis buffer 1 and mix the content by inverting the tube 4 to 5 times. Add 50 microliter of solutiona and mix the content by inverting the tube 4 to 5 times. Then incubate at room temperature for 5 minutes. Centrifuge at 8000 rpm for 2 minutes at room temperature. Carefully discard the supernatant. Remove the excess supernatant by pipetting. Add 1 ml of lysis buffer 1 again and resuspend the pellet in the buffer completely. Centrifuge the sample at 8000 rpm for 2 minutes at room temperature, then discard the supernatant carefully. Add 500 microliter of blood lysis buffer 2 to the pellet and resuspend the pellet by pipette mixing completely. Add 20 microliter of proteinase K and mix the contents by pipette mixing, then incubate at 56 degrees Celsius for 10 minutes. Add 350 microliter of blood magna mix to the lysate. Gently mix the contents by inverting the tube for 10 to 12 times and incubate at room temperature for 5 minutes. Place the tube on magnetic stand for 5 minutes. Wait till the solution is clear. Carefully discard the supernatant without removing the tube from magnetic stand. Make sure that the pellet is not disturbed. Add 500 microliters of blood wash buffer 1, remove the tube from magna stand and resuspend the pellet by pipette mixing for about 5 to 8 times. Ensure complete dispersion of the particles. Place the tube back on magna stand until the solution becomes clear or wait for 1 minute. Carefully discard the supernatant without removing the tube from magna stand and make sure that the pellet is not disturbed. Add 500 microliters of blood wash buffer 2 and gently invert mix the tube without removing from magna stand 5 to 6 times to wash the pellet. It should be surface wash only. Discard the supernatant without removing from the magna stand. Add 500 microliters of blood wash buffer 2 and gently invert mix the tube without removing from magna stand 5 to 6 times to wash the pellet. It should be surface wash only. Discard the supernatant without removing from the magna stand. Air dry the pellet without removing the tube from magna stand at room temperature for 5 to 10 minutes. After drying, remove the tube from magna stand and add 100 microliter of elution buffer. Resuspend the pellet by pipette mixing 10 to 12 times. Ensure complete dispersion of particles. Incubate at 56 degrees Celsius for 5 minutes with intermittent tapping. Place the tube on magna stand for 5 minutes or until the solution gets cleared. Carefully transfer the extracted DNA to a sterile microcentrifuge tube, without removing the tube from magna stand. Make sure that the pellet is not disturbed. Finally, purified high molecular weight genomic DNA is ready.